Okay, thanks for coming, everyone. I'm really excited about the afternoon session that we have set up for today. And I just want to first say thank you to all the main conference organizers for having Giuseppe and I. So I'll just introduce, so I'm Quinn. I'm a postdoc here in Montreal, but I'm going to introduce this person who's standing to my left. This is Giuseppe Gava. He's currently a postdoctoral fellow in the Dupré lab at University of Oxford in the UK. And he's come all this way to give a really exciting workshop and talk today. And I think you'll see a lot of similarities or relationship to the talks that were given earlier today and the sort of analytical approaches that we're going to talk about. But I believe this afternoon is actually the first educational workshop that's been done using animal neurophysiological data. Anyway, about Giuseppe a little bit more. He actually earned his PhD somewhat recently from the Center for Doctoral Training in Neurotechnology at the Imperial College of London, where he also he graduated in biomedical engineering with Simon Schultz. And toward the end of his PhD, he was collaborating with the Dupré lab at Oxford a lot, where he's now a postdoctoral fellow at the MRC BNDU Center. So he's really focused on using concepts from network science, topology, and information theory to understand complex neural circuitry and the dynamics in neural circuits that shape memory and cognition. He had a really exciting Nature Neuroscience paper in 2021 on the data and the techniques that he's going to talk about today. So I hope that you all enjoy what he has to share and that you learn lots from this experience. So I'll let you take it away.
Thank you for the very generous and kind introduction. So today I'm going to talk and guide you through a bit of the type of data that we're dealing with in the Dupré lab, which is it is electrophysiological recordings from most of the hippocampus in mice, in freely behaving mice. So first I wanted to talk about the hippocampus itself. So why this region is actually so attractive to so many neuroscientists. And it's because of three main reasons. So the first one, it's very pretty as a structure. And so what you see highlighted, okay, this red stripe, it's actually all the cell bodies of the principal cells of pyramidal cells, which are the excitatory neurons. And these are the ones that actually project outside of the hippocampus. So they are thought to be actually the information processing units in this network. And because it's so well layered, at different layers, we have different processes of these neurons. We have interneurons, different types, which are basically orchestrating and timing the activity of principal cells. And so because of that, historically, it has been studied a lot just for the basic synaptic physiology. So LFP, long-term potentiation, and depression were actually first found in the hippocampus. Then there is the memory part of the structure, which goes back to the story of the patient HM, which, if you're not familiar with, was that patient that in the 50s, in an attempt to treat his epileptic attacks, he basically had both hippocampi removed. And because of that, he was unable to form any new memories. So that's a severe anterograde amnesia. So he basically was stuck in the same day since the beginning. Thank you.
There we
go. And then the last part, which actually is what we're going to talk about today, me and Quinn, started in the 70s with the discovery of place cells, which are actually, let's say, a functional viewpoint on what the principal cells are actually doing and encoding in the hippocampus. So now electrophysiology is literally just sticking some electrodes and recording the electrical activity in a brain region. They are very nice in the hippocampus. So here I'm just showing a schematic of the usual implants that we have in free moving mice. And it's good because having this structural approach where we have multiple small probes, they basically can span, actually I forgot one thing, they can span the entire CA1 or a good portion of CA1. And why is this part of the hippocampus important or interesting? It's because it's the output, like, let's say, output station of what we think should be the information processing loop in the hippocampus. So we know that the principal cells there have a lot of projections outside. So it's basically thought to be the last relay point of the information processing. So because with these structures we can span, you see the longitudinal axis, these are much better kind of, or in a way efficient way of recording from the hippocampus than, for example, neuropixels, because neuropixels are good when you want to record multiple brain regions that are basically one under the other, right? So you have these very long probes. But in the hippocampus, you would have to basically enter from the side if you wanted, for example, to span a good amount of CA1 or any other regions there. So anyway, given that is the actual technique, what we see are these electrical signals, which usually display some very characteristic oscillatory patterns, like you can see here. This is actually a raw image of what is raw data that is just being visualized and recorded in the recording room. Now, why these oscillations are interesting in itself, apart from being the overall raw signal that we can see in the hippocampus, and that is where we then will pick out spikes of the individual neurons and actually cluster them. But it's also interesting because in the hippocampus, we have two very distinct modes of operation of the network. So when animals are behaving and exploring an environment, we see a very strong, you can see it here, there's very strong theta oscillatory entrainment in the frequency of around 5 to 12 hertz. Instead, when the animals are sleeping, this very strong oscillation is lost, and we have this irregular slow wave, which is interleaved basically by this very high frequency events, which are called sharp wave ripples. And they're very, I think, very famous because it's when these replay events happen. So people think the memories are basically consolidated during sleep, right? Because we do see the same neuronal patterns of activity that happen during exploration. We see them again during sharp waves, but these events are very short. So basically, it's like a compression of what would actually be naturally encoded. Now, all of this is very interesting, and I think is very much one of the, let's say, very cool aspects of electrophysiology, that it allows you to record the fine timescales of actual neural activity in terms of voltage, which we believe is really the main medium, right, of their communication. And here I just wanted to flag work from Vitor. Is in my lab. He's a very smart and cool guy, and basically he had this very nice paper where he showed that on top of what we see as normal theta during exploration of the animals, there are actually much more complex oscillatory patterns that kind of distinguish which mode of operation, even further than just normal theta, is actually happening in the hippocampus. And so it kind of starts defining the state of the network a bit more. Now, I'm not going to talk about that because I actually don't use it. I just thought it was it's a cool study, and maybe you want to have a further look later on. Now, what I deal with is actually spiking activity of neurons. So the data I use will be mostly like this, where I have a lot of up to 100 simultaneously recorded neurons, which are spiking. Now, these spikes were extracted from the raw electrical signals by doing a high pass filtering, and then there is template matching to find the putative cells. But anyway, and in blue instead, you see, for example, the only real LFP signal. So this oscillatory pattern that I care about right now, which basically tells me whether the state of the network is reflecting an entrainment of the animal, so an engagement of the animal in exploration, or whether instead it's just resting and not really being engaged in the task. Now, I'll go and talk a bit about actually my work. So, given all this activity that is happening in the hippocampus, there is a lot of literature suggesting that what is actually represented there by the population activity, it's somehow the overall context or experience the animal is going through. So with the idea of place cells came the idea of spatial maps, trying to basically represent, in a way, or spatially map the environment. And now this also goes a bit further on, so also describing the actual context, maybe even the mental state that the animal is going through at that moment. Anyway, in this work, we wanted to study how do novel memories basically gets integrated in a previously existing network of knowledge, basically, of previous memories that the animal already has, right? So to do that, we go through this task. I mean, we have animals going through this task, which is called condition place preference, which is CPP. I'm very sorry that you probably won't be able to see a lot of the labels.
They should be able to see them on.
Okay, on the computers,
Can you all see the figures better on Crowdcast?
I guess, because more small fonts is going to come. But anyway, okay. So these animals are basically first exploring a familiar environment, which is dubbed FAM1 there, and then they go through this place preference protocol, where we first test the naive preference of the animal for one of the two environments. Here are called knob 1 and knob 2, which are connected by a bridge. And so the animal can freely explore both of the environments. Then we pair the least preferred environment in the successive session with sucrose. So basically, the animal will find sucrose at different locations in the environment later on when it explores it. And in the session afterwards, instead, we pair the most preferred environment with water. And what we're trying to do is just making the animal change his mind, basically, in which is the preferred environment. And in fact, when we test them after an hour in this CPP test session, which is the same as the pre-test, so the two environments are empty, so the animal is just exploring, we actually see that they basically always do change their preference for the environment that has been paired with sucrose. Okay, then after this test, we take the animals back to the familiar environment because we are interested in seeing how the representation of, how a very familiar representation, because this mouse has been seeing this familiar environment all his life, basically, has actually changed after the condition, after the task. Okay, so here you can see, again, another raster plot, so where we see the spiking activity of some neurons. You can't see it, but basically, at least I can't there. So I'm only considering principal cells. So what is also interesting about EFI's recordings is that we can have a very good guess, at least in the campus, at whether the neurons that we pick up are principal cells or interneurons. And as I will show you later in the demo, principal cells are the one showing some information encoding, and interneurons are instead very active, and they seem only to basically regulate the activity. So in this case, I wasn't interested in looking at them. So now, and given this very, population activity, what I wanted to do is basically recapitulate the relationship between neurons, between the activity of neurons, by looking at their coactivity measure. And this is basically a simple Pearson correlation of two spike trains only during active times, so when there is theta and the animal is moving, and basically is very similar to the functional connectivity metrics that you saw this morning. Just instead of being voxels or brain regions, we now have individual neurons. And so, for example, here we have 68 principal cells, and I'm trying to recapitulate how they are being active with respect to each other. Okay, so down there, actually, you can see a submotif of nine cells, and how actually they are basically changing their co-firing during the novel task, during the CPP task, and how the patterns are actually being taken back at the end, recovered basically, when the familiar environment is seen again. Now, to characterize, let's say, the topology of this co-firing activity, I take three simple measures from graph theory, which are the co-firing strength, which simply summarizes how strongly a neuron is coactive with all its peers in the network. Then there is the clustering coefficient, which instead measures the amount of coactive triads, so triplets of neurons, which are coactive with respect to each other, or fire together, basically. And the last one is instead the geodesic path length, which in a way is a proxy of how well does the neuron, how easy is it for one neuron to communicate on average to all the other ones in the network. And so what we see is that during the task, basically all these measures increase because there is a heightened firing. So the cells do actually couple more as more information is being processed, let's say, by the network. But what was also very puzzling is that when the mouse is put back in the familiar environment at the end, the green section, these measures don't go back to baseline in this case, but they stay heightened, showing some kind of hysterical dynamics, which is suggesting the previously formed representation, right? So memory that was already there has been changed by the encoding of some new information. Now, to kind of look a bit more into that and to control some factors, what we did was to basically actually set up different tasks. So for example, here, which we called novel context only task, the animal is always exploring the familiar environment at the beginning and the end, but then is just seeing 4 times the same novel environment. And what we see is that actually the same co-firing dynamics are actually happening during the exploration and the novelty, the novelty encoding in the task, right? But then they go back to baseline at the end. Same thing when the animal is now basically going through the same task, but there is no association with food. So we're not really changing their mind. So when they go through the two middle sections. Now there is no sucrose of water. And again, we see an increase in firing, a changing of firing basically during the encoding, but it goes back to baseline at the end. And a similar thing when, well, basically now we have only familiar environments and there is pairing, but there is no novelty component and we don't see anything. Yeah, and this is just to show that basically in the condition place reference task, we do see this hysterical dynamic. Now, neurons in the hippocampus are famous for their place cells, right? So the first thing we did to actually characterize this change in the representation was to look at place cells. And as you can see in these examples, many cells do, we say, remap, so they change their place field when they encounter a new environment, okay? But then when they go back to the re-exposure, they basically show again the same firing field, suggesting that basically, and here is a quantification, that actually there is an reinstatement of the familiar map, which is basically it's a kind of a byproduct of the fact that the as I showed in this little snippet here, we do see the same co-firing re-emerging at the end. So we're not surprised basically to see the same maps reappearing as well. And this is because in a way the spatial map or place field of cell is measured over like 20 minutes of exploration. So it's a very crude approximation in a way of the actual, possible function that this cell is taking. Now, to take a closer look at at the structure of this firing and these representations, here I took basically a distance measure between two co-firing matrices, and with the co-firing matrices are basically representing the actual environments. And what you can see down here are basically this 6x6. It's summary of the distances of the net of the co-firing pattern of the network in each of the 6 sessions the mice went through in each day, and they are basically the distances measured in a pairwise fashion. So we get 36 altogether measures, but they are symmetric. They should be. Now, to visualize actually the information that is present in these matrices, what we can do is we can just apply a simple PCA in this case, and we end up actually visualizing where do the representation of each individual session actually lay in a lower dimensional space, because as you can see from naked eye, the two familiar sessions end up being close together, for example, and the 4 sessions in the CPP are actually very different from the two familiar ones. But now, and now basically I show you what we see when actually we consider all of the recording that we had from different mice together. And it's interesting because when we apply then this PCA on these distance matrices, we find one main axis, which is differentiating the familiar and novel context of the environment itself. Along PC2, we see a separation between the 4 individual experiences in the novel environment. And along PC3, we have this, we have highlighted basically difference that we saw with the hysteric dynamic with the co-firing basically. Now, it has been a lot of data, a lot of, graphs. So we can move to the actual notebook where we can have I hope a better feeling for what these data actually are or look like. So this is the collab notebook that you should find from the GitHub page. So on the README file, I think in the third from the last line, there is a simple link to this same notebook. Now you can either, yeah, well, Jan can help you, is over there on the left. And
so if you're having issues finding the notebook or anything, you can also come around, so just let them know.
Cool. Now you can copy this notebook maybe to your drive if you want, but or you can just, if you want to do any if you want to tinker with it and play with it, or you can just run it as it goes from here. So I will go through it myself. So what we're doing here now, we are basically, yeah.
If I just zoom in for the online document, it's small.
Oh, okay. Okay. So basically now we just, can you say yes? We're just loading basically the GitHub repository onto Google Drive so we can access the data.
You could like just accept this.
I'll do that. I'll do that.
Okay, maybe you'll continue this. Yeah.
Okay.
And also,
I think that it was trying to make a copy and
Yeah.
So
I was trying to make a copy.
yeah, so just let
Yeah, I
me
was trying
log
to
into my
make-
email and I can take care of that.
And also light mode is better for like a
Yeah.
social audience. Just ensure it's in light mode and zoomed in.
Yeah.
Sure. Thanks.
Okay. So now we should have access. So now it's doing its things. But okay, good. So I load the data now because I already had it downloaded. It didn't actually download them, but it should be a very light process. So I guess if you're also doing it, should be done pretty quickly. Now we're going to import the usual libraries. I'm actually going to use two custom libraries, but all the functions are also present in this notebook. So in the GitHub repo, there is one utilfunc.py, which is basically containing all the custom functions. Now, but I did copy them here if you want to have a look later. So basically, the first ones are really just to process the neural data that we're going to look at. So we're going to have to bin basically the spike trains to have a common time framework for all of the neurons together, so that we can actually take population slices of activity in a way. This function it's a Gaussian smoothing function, so nothing crazy. Then get actmat, that would be a theta, is basically using the theta information to extract basically the activity matrix only during active times. The ISI is instead the interspike interval, and I will show you later what actually is. It's just a way of characterizing or summarizing the way that cell is active, so its activity. And the place map is just, yeah, it says in the name. So let's go on and set the parameters and load the data. Okay, so in this recording, we have 74 putative principal cells and 6 putative interneurons. Now we're going to have a look at what their activity looks like. Now we are also going to load the spiking activity, and this is really just a technical thing, a detail, but basically the way we store them is as two in two big files. One tells us all the spiking events that happened, which is going to be the res variable here. And the clue variable instead contains the cluster to which we think that spike belongs to. And now the clusters are basically units. So actually individual neurons, right? So in this case, we'll have 80 basically clusters because we have 80 cells altogether. Now, what I'm simply showing you here. Oh yeah, I didn't run it. Perfect. Okay. Basically, simply some raster plots. And I want to show you what interneurons look like as well. So here I'm just showing you 10 seconds of activity. And as you can see in the plot from the left, we have all the units, and on the right, only the putative principal cells. Now the interneurons are actually only 6, but we have 3 very fast spiking interneurons, and which are the ones that are basically a black line because they're always active, or at least a lot more than the other cells. Okay, so now what we can do as well to kind of have a global picture of what's happening in the population is to actually plot the distribution of the fine rate of all the units. So interestingly enough, we say we have a log normal distribution of fine rates in the brain. So we have a log axis on the x, right? So it's normal, but only if we plot it on log scale. And this is basically the hallmark of a distribution which has the majority of units being very lowly active, so basically silent, and we have a small minority of very active neurons. Okay, and so as you can see, if I'm plotting all the cells, we have this 2 gray steps going. Basically, the last step is going up to 100 hertz. It's probably only 50, but because of the binning, we see it like that. And once we take them off, we actually end up with less than 10 hertz, which is the kind of maximum fine rate we know that principal cells usually have. Now, to tell whether a cell is a principal cell or interneuron, I guess you're not too interested, but it's mostly based on rate, okay, and also on the shape of the spike they have. So when we look at this very fancy electrophysiological trace, we do see many spikes, and the way we know which spikes belong to which neuron is by basically triangulating, right, the same signal measured with different electrodes in the brain. And if we see the same shape of action potential, so spike, that is telling us that the same neuron actually fired that spike. Okay, so now what I'm going to show to you here, it's simply another. I think we didn't run these functions. Yeah, didn't run these. Okay. So now we have the functions, and we can look at the ISI. And I want to show you these ones because, again, they hallmark this, what is interesting about principal cells in general. So here again, you've got a log scale on the x-axis. And what is interesting is that principal cells are bistable. So this tells us that basically they have a preferred state at which they become very, very active. So they actually emit bursts of spikes, which have less than 6 milliseconds of inter-spike interval between each other. So basically, yeah, just a very loud burst in a way. And but the majority of the time, these principal cells are actually very quiet. And so they will spike like 1 spike every second or even less on average. Instead, interneurons don't really have two defined states. And so they are always basically, at least we can't really will identify different states in which they are, because they are always kind of orchestrating the activity of all the principal cells. And to follow the very fast rhythms, they have to basically, they are making the rhythms themselves by being so active. Okay, so now what good principal cells in the hippocampus are known for is that when the animal is basically exploring an environment, and here you can see the trajectory, we see that these neurons actually show place maps. So here I've chosen a selection of them. Actually, it's the first, I think, first 16 principal cells where you can see, some tunings. So some narrow spatial tunings, like in cell number 4, for example, is very pretty. Cell number 9 is pretty as well. Cell 3. But then if we go on the last line, I actually chose 5 interneurons, and you can see that their firing is not really place specific, so they're always active, and therefore they don't show any place map. Now, what has been used in the literature to describe the amount of information present in or encoded by each of these units is basically the mutual information, which comes from basically the Shannon derivation in the 40s, which then later Skaggs basically simplified for place cells. And what I'm showing you here is just the distribution of information across the population. And as you can see, basically there is a small trend with fine rate, because basically here we're just measuring the rate of information, so bits per second, So you can imagine that if quite a fast firing cell is also informative, it will have a higher rate, right? But then these 3 last points at the bottom are basically 3 interneurons. So they're very fast spiking, but again, they don't really encode that information. Another measure that was proposed is basically trying to normalize the rate, the temporal rate, but the actual firing rate of the neuron. So giving an estimate of the bits per spike. But this is, I'm not a fan of this, because it has a very, very strong anti-dependency with fine rate. And so you will find that the cells that only fire maybe a couple of spikes during a session actually have a very high information, just because their fine rate is very low. Okay, so now down to actually generating the co-firing networks that I showed you before. So my approach is to, and you can see it, for example, here in this first paragraph, I'm just actually, well, I didn't actually pinpoint where I was extracting all the spike trains, but given the rest include, then it was easy to basically match each spikes to each individual cluster. And now we want to have a representation which has the same temporal framework. So we have to bin the spikes. And to do that, we simply basically use this bin spikes function, which I wrote, but it's simply an histogram function. Okay, a bit fancier, just put them in the right format. But basically giving the bins, which is basically an array going from 0 to maximum time in 1250, basically the sampling frequency of the row that I showed you before. We basically divide that in bins of equal size, and then we just count how many spikes basically these neurons fire. And so I don't think I actually have well, if you want, you can simply do a PLT im show on a part of ActMath, and you will see that this is really just a matrix. We have time on one axis and number of neurons on the other. Okay. Now that we formatted the activity, we also then load the theta information, and now this comes as just another matrix where I have number of cycles of theta and some information basically when there is the peak, the trough, etc. So the, let's say, salient points of each cycle. But what I'm actually doing right now is just taking basically all the samples when there was some theta happening. And so in this, how did I call it? So in this theta variable, it's basically a Boolean vector, right? That will then index my ActMath variable. So when I'm later on, so I first also extract the active times when the animal is moving, okay? Then I put these two together to get basically an overall active Boolean vector, which is this theta active, and then I can just simply index it. Index ActMath when it's active is basically the overall ActMath during the active times. Okay, so now I can also just use these bins defined by the theta cycles to actually format the activity in a different way. So instead of having individual samples at 1250 hertz, I can actually use the whole cycle as just one sample. So it's like a physiological binning, basically. Instead of being of equal temporal length, they are kind of equal temporal length to the brain, because we know that each cycle should correspond to some kind of temporal window for the hippocampus to actually process information. Okay, so what I'm doing here in the next cell is actually displaying these graphs. Actually, I'm obtaining them first. So now I'm obtaining. I think this is a bit of a. I think I didn't run a particular cell first. Sorry, I think it's this one. So I don't want to go too much in the weeds with that, but basically this might be of interest for you later, maybe. We were here, huh? I just wanted to compare basically 3 different ways of obtaining it's not defined. I'm just gonna take it from the function. Should be fine. So I have a backup py file which should have all these functions inside, so it shouldn't be a problem. Yeah. So I just want to compare them to show you what they look like, but I think we might be short in time, maybe.
Yeah, there's another 8.
Okay, so we'll skip this, but you can have a look at that later if you want. What I'm doing here instead, sorry, this one, is basically showing you some basic analysis of the actual work that I showed you before. So summarizing the firing strength, clustering coefficient, and other topological measures in this data. Now, yeah, I realized this is a bit over time, so, and apparently I have again to in Set the functions, okay. But basically, I also okay. And now, of course, once more. And another one is. Okay, so basically now I'm just extracting measures from these graphs, and now I should say that basically one of them is the same recording that we had a look at now. The other ones are basically 3 recordings from the different tasks that I talked about before, and we're just summarizing basically some of the measures to describe this firing activity and structure. Now, what I'm going to show you here is basically, I really have to, I think, yeah, I think it was that, but we are doing it fine. Yeah, now this is all defined. So basically now I'm just showing you how to obtain the same panels that I showed you before at the beginning. So to visualize this cofiring matrix and the graph that we can extract from it. And now later on, when this is run, also here, I'm showing you instead the sub network of graph of just 9 neurons actually, where you can better appreciate the dynamics in their cofiring structure and how does it change as the session, and so the environment changes as well. Okay, but I see that this is, oh no, might be, well, it's not the prettiest of the graphs, but basically it's what we saw before. Now, and the position of the nodes is stochastic, so changes every time. Now, what we did wrong, we forgot to put another call. Yeah. And now this one should be much shorter. And yeah, just visualizing the subgraphs. I thought it, yeah, would be pretty and interesting just to appreciate basically how the cofiring patterns that we see in the first familiar environment do change through the exploration of these novel environments, and then they come back at the end. Now, I won't bother with this. I wanted to instead show you so one example of the topological matrix that I was talking about before basically summarizes the similarity of the cofiring patterns across the day for the different sessions, okay? And basically when we then use PCA to have a look at what they look like, here we basically obtain some 2D dimensional representation where the colors actually are representing the sessions, and I guess the main point being that when we look at the dimensionality of the representation, because in the CVP task there is a lot of variability, so each cofiring network is actually processing more information. We do have a higher dimensional representation, which is a hallmark of it is basically this almost linear decay of the explained variance in the PCs. Instead, in the other task, especially for example in the familiar reward, where there are 2 familiar environments which are explored, 2 times, we see that basically PC1, which is the one just representing their difference, is explaining 90% of the variance. And you can have a look at the other ones if you want. It should be hopefully well described. I think I won't bother you with the rest of the talk. I really was just going to kind of finish off the work that within this paper, showing how these actual different axes seems to be related to the activity of different population of neurons in the hippocampus. And so we see that, for example, low activity cells, so the big majority, part of them is actually the one being recruited to integrate this new memory into the previously existing network, and is the one actually carrying the differences that we also saw in the co-firing hysteresis at the beginning. So I'll just go to my summary slide, basically. Then again, thank you, Quinn, for inviting me. And yeah, just want to say thank you to, my lab and collaborators. And that's all. Thank you.
Hey, so my name's Quinn. Thanks again to Giuseppe for the great talk on EFA's data before. The reason why I asked Giuseppe to come here is because in the work that I'm doing in the Brennan lab with other postdocs and students, we have very related techniques analytically in some capacity, but we're applying these techniques to a different type of data, specifically calcium imaging data in freely behaving animals. So I'm going to talk about, in this part of the session, how we can leverage representational similarity analysis to quantify the dynamics of cognitive mapping in the hippocampus when environments are changing shape. So you can imagine how that bears a lot of similarities to the types of experiments that Giuseppe was talking about earlier. But we're going to be using a different technique that affords different benefits than electrophysiological recordings usually do. So I know actually really just one of you in the room, but for most of you who don't know me, before I started doing anything in neuroscience, I fell in love with studying the brain when I was an undergraduate student in Squamish, BC, at this tiny school in the mountains called Quest University. And after I finished my graduate degree there, I went to do graduate school at the University of Lethbridge in southern Alberta, not so far from where I mostly grew up in Calgary. And there I was studying the systems level organization of memory with Rob Sutherland and Rob McDonald, and mostly doing behavioral neuroscience. And while I was there, I learned about some techniques where you could record hundreds of neurons simultaneously and look at their representation in memory tasks or navigation tasks in freely behaving animals. And I thought this was the coolest damn thing I've ever seen. And so not long after I learned about these techniques, I met Mark Brandon at a CAN conference here. And he invited me to come to his lab, and I've been here since May 2019 in Montreal. So long before I was into neuroscience and this whole journey happened, I actually grew up in the US for a while, and specifically in Southern Oregon. And I wasn't really into team sports as a kid growing up. Instead, I turned to skateboarding, and this was my absolute favorite place on earth when I was 7 years old. This is the Ashland Skate Park in 1999, when I would have been around there a lot. And just summer before last, actually, I still skateboard, as a matter of fact. And summer before last, I got to go back to this same place where I grew up. And while I recognized the park still, like it was in many respects in my memory, many things about it were different. The concrete had eroded in many spots. There was new graffiti. There were bigger trees than before. They had gotten older, and a lot of the obstacles also felt smaller because I had also gotten bigger. But despite this, I was still able to navigate this space and actually take the sort of same paths and lines through the park and do some of the same old tricks I used to do when I was 7 years old. So the reason I'm talking about this, and I've been talking about skateboarding and growing up for the last few minutes is because I think it points to a thing that really interests me about the brain and behavior, which is how does our brain map a familiar but dynamic or changing environment over a protracted amount of time and experience? How do you return back to these old familiar spaces that have changed, our world is constantly changing, and still know how to navigate them and achieve your goals within those spaces? So the way I want to start talking about this is really with the first part of this question: how does our brain map out space, spaces that we navigate and explore? So I'm going to talk about some things in overlap with Giuseppe's talk, but the best place to start is probably here. So this is a place cell. We already talked about this in the last hour or so, but the discovery of place cells and their initial reports were happening in the 70s with this seminal work, The Hippocampus as a Cognitive Map, describing a whole theory about how the brain creates maps of space based on these types of neurons in the hippocampus. And specifically, this is a CA1 principal neuron, which we are talking about, of a rat. As the rat moves around the box, and in a restricted part of this space, this neuron will fire, as indicated by these black dots. So when you average over time, you get the neuron expressing this sort of spatially restricted firing field or a place field. And many, many, many CA1 neurons express this property. So what happens when you record place cells that have place fields, but you change the environment? So later in the 80s, John Kuby and Robert Muller started asking this question, recording place cells, like the one cell shown in the bottom here, when they changed the environment the animal was in. So if you record one place cell, a hippocampal principal neuron, in one environment, let's say it's a big circle with white walls, and it's in some room off in that direction. And you take that rat, that animal, you bring them into another room, and you put them in a big circle, the same exact shape, but now the colors are different in the chamber. Things have changed, but it's really the same geometry. You find that the place cell remaps. So the firing fields of that cell, its spatial map, completely changes. Now, this is what happens when you change the rooms. It forces this global remapping to occur. But later it was found from Laura Kolgen and the Moser group in Norway that if you instead keep the animal in the same environment and you start to change the colors of the walls, for instance, or some sensory information, there's a more nuanced effect that happens. A neuron can fire in the same location but change its firing rates instead. And this is often referred to as rate remapping, but collectively this phenomenon altogether is referred to as hippocampal remapping, which Giuseppe was also talking about earlier on. And there are many, many things that can cause remapping of hippocampal place cells, but one of the strongest determinants that we've learned about over the last 40 years is the geometry of an environment. This is a really strong determinant of mapping in the brain, at least in rodents. So in this experiment from John O'Keefe's lab and Colin Lever in 2002, they were recording rats and then changing the shape of an environment specifically. So what they found is when they recorded the same neurons, which are shown vertically here, a lot of the neurons seem sort of anchored to the geometry of the environment. They get pushed and pulled by this geometry in this space. So if a place cell is active in sort of this region of the environment, right along a border of this circle, and then you change the shape of the environment so now it's a square, it gets pulled over into the corner of the square. However, when you give the animal more and more and more experience, let's say 21 days of experience, and you do the same experiment, the changes become much more dramatic. So now you get neurons that can go silent when you change the shape of the environment. So the remapping can become more global. However, there are still some neurons that seem very anchored to sort of the border and the edges or the shape of the environment. Okay, so we know that we now have a model system where we can record neurons that map out a space. They change in really interesting and dynamic ways when the world around us changes. But one issue with these early studies, or one limitation, is they were using techniques where you couldn't hold on to the same neurons, record the same cells for a long period of time. But in a system that we know has an important role in memory and encoding experiences, we might want to hold on to the same neurons and track them as the animal's having the same experience over a protracted amount of time. So neuroscience recording technology has come a long way in the last 20 years, especially in the last 10 years. And now we have techniques like calcium imaging, which I'm going to talk about today, that allow us to record the same cells and then track them over extended periods of time using optical methods. In parallel, there are other techniques that are emerging in electrophysiological recordings as well that allow us to hold on to neurons for maybe a little bit longer. But they also have different limitations, some of which Giuseppe talked about earlier. So we can start to get at this part of the question. How does the brain map this changing environment across a protracted amount of time? And in particular, I'm going to focus on this technique in the upper left, which is calcium imaging in freely behaving mice. So I'll go through this just briefly. But you're probably all familiar with many of these techniques that afford different cellular, spatial, and temporal resolution. They have all different sort of affordances experimentally that we might be interested in. EEG and LFP recordings, for instance, allow us to record from the same brain for a long period of time, but they don't necessarily give you single cell resolution. We can use some optical techniques that give us cell type specificity, and we can use some other freely behaving optical methods like fibrophotometry that allow us to do recordings in freely behaving animals over long periods of time. And miniscope recording that I'm going to talk about today occupies this space somewhere in the middle, where we can record a bunch of neurons with single cell resolution, and we can track them over a period of weeks or even months. So the concept, the approach to doing this is we could take a big complicated wide field microscope, fluorescence microscope, and really just break it down to its bare bone essentials, which are a camera to collect image data, some optics to bounce around light and hit a sample and then collect light from that sample that would be fluorescing, emitting some sort of neuronal signal. We have an illumination source and then a controller inside here that's going to control all of these components. And we can miniaturize all these things with modern printing technology. So in 2011, there was this Nature Methods paper from the Schnitzer group describing exactly this, miniaturizing these critical components of a fluorescence microscope. So you have a camera, some optics, an illumination source with a small LED, and a controller that could be integrated with the camera on a small printed circuit board or offline with a computer to image within the brain of a freely behaving mouse, because the camera is really small and could weigh between 2 and 4 grams. So a mouse could actually run around with this on its head. So there's also open source versions that we're using in the Brandon lab that are very affordable and have been simplified a little bit, so you can do this without a huge startup package in your own lab and a little bit of technical know-how. But we can peer down into the brain of a freely behaving animal and record fluorescence activity of individual neurons. Pretty cool. So the general approach to get neurons to fluoresce, and these are some data that we've recorded in the lab that we'll be playing with today. The general approach is we can inject a viral construct into our target region. And here we're going to focus on CA1 in the hippocampus because we know there are place cells there. And that's what we want to study. So we'll inject a viral construct that will express a calcium indicator. And whenever neurons are spiking, well, most of the time when neurons are spiking, they're going to let a large influx of calcium occur, and then we can image the soma of neurons that have these calcium influxes. So we can actually identify hundreds of neurons in these recordings, up to 1,000 in a single animal, and we can track them over a long period of time. Here's 200 CA1 neurons, and they're rate maps. We've been staring at these a lot for the last couple of hours. And you can see that all these cells, or many of them, have these restricted firing fields. And with this technique, 10 days later, we could look back at the same neurons in the same environment and ask, how similar are the maps at a delay of 10 days? And you can see in many cases, the place fields seem pretty similar, but not in every case. So this starts to really allow us to address a lot of questions about long-term representation in the mouse brain. And that's what we're going to get into today. So this is the question we're after. Right now, I'd like if you could make sure you have the Colab notebook for this session opened, and please start running it now, specifically the first kernel, so that you can download some of the data, because it could take a couple of minutes to download. So if you go here, this is the notebook that we're after. You want to run this first kernel, and it took me 1 minute and 34 seconds to download this data. I think there are 900 neurons over a month of 40-minute daily recordings that you're going to download in here. Okay. So if you could just run that kernel and get things rolling, that would be great. Okay, we need to get more specific in how we're going to evaluate this question. Over the decades, or almost century now, that people have been studying this phenomenon of cognitive mapping, how our brain maps out of space and how we navigate it, there's been many streams of hippocampal research and theory and proposals on what this brain area does, how it does that. So there's this one stream that we've been kind of focusing on that's really on spatial navigation and spatial representation. But there's this other history, very rich and important history, of hippocampal research on memory. And Giuseppe talked about that earlier. And more recently, there's been some theories that have unified these different sort of streams of hippocampal research into these more generalized models of cognitive mapping and memory. And actually, Patrick talked about a little bit of this in his talk today as well. So here's two such examples of a couple of theoretical models that produce place cells. So this is a boundary vector cell successor representation model. So this is a model that comes from reinforcement learning literature in some respect, that it uses a successor representation to model the hippocampus as a sort of predictive map. This is another model that actually Patrick was referring to from the Barron's group. And this is the Tolman-Eichenbaum machine model that can produce a lot of these sort of grid cell or place cell type dynamics that we see in the hippocampus. And they explain some of the memory functions of the hippocampus as well. But there's no consensus on how we should actually evaluate these ideas and compare these theoretical theoretical claims with empirical observations that we can make about actual hippocampal mapping. So how can we compare the way the brain maps space with the way that computational neuroscientists are claiming it does? And how do we know how similar or different these models are from each other? Because sometimes the claims, I mean, to me, look pretty similar. The one very promising approach that you've already been hearing about today is to use representational similarity analysis. And so to sort of explain this a little bit further, the idea here is that we have some assay of neural activity in the brain, maybe calcium traces from individual neurons or spikes from individual neurons. And we present a bunch of conditions to this brain. And we have a dependent measure in each condition from this assay. What we can do is we can measure the magnitude of these differences between all of the conditions. So you have A versus A, B versus, B would be along the diagonal here. And then all the comparisons between and across the conditions are in this off-diagonal space. And the reason why we're interested in doing this is because we can compare this representational similarity or distance matrix to the same sort of distance matrix in the same conditions with other types of assays or other brain areas, other organisms, or a model system. So you can imagine recording the same assay in a different brain region, present the same conditions, ask how different they are, and we could compare this representation with this one. But we could also generate this representation with a model system, say your favorite neural network model. So this allows a lot of flexibility to compare and contrast empirical observations we're making with the theoretical models of interest. So my collaborator and I, Alex Kyneth, who just is starting her lab right now in Chicago at UIC, a very talented, previously undergraduate student, now graduate student, Erica Chianferrano, and Mark Brandon, we asked how could we design an experiment with this analytical approach in mind. So let's start with the classic mouse in a box approach. And this could be a rather large environment, 75 by 75 centimeters, a big open field. But what we're going to do is imagine this square space like a tic-tac-toe board. So there's actually 9 partitions, 9 subspaces in this environment. And we could block out different parts of this subspace, these partitions, to create a bunch of unique geometries. And we could get really creative with this. And what we did is actually we created 10 different geometric forms that we would record on separate days while we're observing activity in the mouse hippocampus, and the mouse is running around this space. So on the first day of the experiment, the animal would go into the square environment. And then on the next day, it would come back to the same space, but we'd change the shape. So let's say for this animal, it would be the cross form of the environment. We'd keep on changing the shape of the space. We have a C here, a donut shape here. And then after they've gone through all of these geometries, we repeat the shapes of the environment for a total of 3 sequences, always bookended with these squares, right, as a sort of undeformed environment. So we've recorded actually 7 animals, but here's 6. With these animals, this is the 182 here, we get hundreds of cells that we can record on each day. And so we're using this classic sort of viral vector approach with calcium indicators, GCAMP6F specifically. And we'll take these videos and break them into spatial footprints, which show us where the individual cells are that we've recorded, and these calcium traces, and we binarize them down here. We track the animal's behavior using deep lab cut. So there's 31 days of recording. The square appears at intervals of 10 days apart. So we can stably track the animal's trajectory in the environment across time. So here's the trajectory, and it's color-coded by the animal's velocity. And with the neural data and the animal's behavior, its trajectory, we can create our beloved rate maps, right? So here's some rate maps from a single neuron, so the first neuron out of 853 neurons, that are recorded across these 3 sequences. And there's actually another square here, I just haven't included it. But you can see that this cell, across all of these, or many of these deformations of the environment, has a very similar place field. Some of them, like the cross, it sort of disappears and goes crazy. But then it's often appearing in this lower left-hand corner. This cell on the bottom here has this field that's biased towards sort of the bottom right-hand border of the environment. But we also see neurons that do really interesting things in CA1, like this one here. And it increases its firing rate with some more experience in the space. And then can have double fields that often favor a sort of right-going wall. So you get these double fields that get pushed and pulled around by the space. Okay, so how can we use maybe rate maps or other assays of neural activity with representational similarity analysis, and then compare the representation in the mouse hippocampus to theoretical models? So we're going to do the demo now. I don't know if anyone knows Todd Church. Well, I think I wanted to keep that open. Here we go. So hopefully your data is downloaded. This particular notebook that I have up is, this is going to be really hard because I can't see down here. One sec. There we go. So this notebook is to be completed. In the GitHub page that this comes from, there is a completed notebook if you want to just walk through that. But there's actually stuff that you have to enter for this to work. So we can run these first few kernels. This is just a depiction of what we're trying to achieve today. So once you've loaded in the data from the Dropbox, you can run this kernel here to actually put it into a Python dictionary. And I have some helper functions that, if you're so inclined, you can go dig around and look at some of my messy code that I hid away. So this is going to load the data into a dictionary. I'm just going to take out some data we're not going to use to save a little bit of memory. And once that loads up, we're actually going to just get an impression of the way you can index out or pull out different values that we want to use for this analysis in this dictionary. So the animal I defined up here, it's this animal QLAC CA150, animal 50 of a bunch of recordings we did. So the way that you want to pull out the values from this dictionary are to just throw in animal and then specify the field. I think this is, yeah, the position will be like this. But this is really just to give you an idea. If you want to play with the data yourself or call it up later, you know how this is roughly organized. But you could also just print off the keys as well. So yeah, you can use this string position after animal in the first field to pull out the position data. And then to get all of the rate maps that I've pre-computed, you can put in maps. And there are 2 forms of rate maps nested in there. One of them is smoothed, and then there's also an unsmoothed version of the rate maps too. The smoothed ones we've just gone over with the two-dimensional Gaussian kernel and smoothed the fields a bit. Oh, I promise I know how to spell. Okay. There's a lot of data here. There's 31 days here of recordings, 942 cells we've recorded from, and there's almost 72,000 frames per session. Okay, pretty big. And then XY position, the two-dimensional position of the data or animal, is 31 days, X and Y position, and then the same number of frames because the position is aligned.
What is the sample frequency?
30 hertz. Yeah. And then the rate maps have a slightly different dimensionality. It's 15 by 15 X and Y bins, spatial bins, 942 cells, so a rate map for every neuron, and then 31 days of them. Yeah. This is miniscope data. Yeah. So also I have answers in here if you need to take a peek and you just want to quickly get through them. You could uncomment this with the handy command / hotkey for me on a Mac and run those kernels if you'd like. Okay, so I want to first just plot a few minutes, the first couple of minutes from a few example sessions so you get an idea of what the data look like. I'm going to pick all of the square days, which are the first day and then every 10 days later. So 0, 10, 20, 30. And then this function I have is going to plot the traces. Can't really see them there, but you should see several hundred neurons and then very dynamic X and Y position data that's we have aligned to the neuronal time series. So this serves as the backbone for building all of the rate maps. So when we're building a rate map, we take the animal's position, and whatever firing is happening in the time series of neuronal data we have, we assign that activity to the position the animal's in. Okay.
So could you explain that?
Yeah, so when we're building a rate map, imagine the animal's walking around in a grid. So we have a spatial binning of the environment. Here it's 15 by 15 spatial bins. So each bin corresponds to 5 centimeters squared. And so we take the animal's position data, and then as it's walking around in this grid space, we add in the activity of each neuron to that bin, and then we just average over time. And that's how you build a rate map. Yeah.
So that may be just a good question, but wouldn't there be some sort of a time shift maybe?
Which, sorry?
Some sort of a time shift maybe?
We align the data with 2 sort of frame maps that we have. So we have a bunch of times when we've registered every frame of the animal's position data from the camera of its behavior. And then we have all these times when we registered every frame of the calcium data, and we use that to align them in time. Yeah, and it's a 30 hertz recording. So the first thing I want to do is actually just grab some firing rates of these neurons. Firing rates is maybe a little misleading in calcium data because really it's we've had calcium traces and then we binarized them, and then we're sort of inferring a degraded signal of electrophysiology that's causing the fluorescence in these neurons. But nonetheless, let's see what it looks like. So I have this little for loop started here. So I just initialized the mean firing rates in this zeros matrix. And what we're going to do is just average across time. So if you actually just want to print real quick, traces. So which axis do you want to average across if you want to get the mean firing rate of all your neurons? So 942 is the number of neurons. So in Python, which is 0 indexed. The number of frames is this 71,866. So if you want to get the average firing rate of every cell, then you could say traces dot mean of the first or the one axis. It's hard to say first in Python. Of this axis here. So you could average through all time in every session and get the mean firing rate of all the neurons in every day. Okay. So let's just take a quick look. This is a sort of distribution of firing rate or activity rate, you could say. In general, in this case, it's kind of increasing with experience in this environment. We can't say for sure how much of that is due to the way that we're recording neurons optically, if there was maybe a change in the fluorescence. There are ways sort of with the pre-processing that we try to control that, but we're not going to bet the farm on changes in firing rates just averaged over the whole session. But we can still look at relative firing rates among all the neurons over time. But you can see that this is a very heavy-tailed distribution, right? So like Giuseppe was talking about earlier, most pyramidal principal neurons in CA1 are quiet a lot of the time. Then you get these moments of high firing rates. So this is kind of what you expect, this sort of heavy-tailed distribution across days. So here, I'm just going to grab some example rate maps that I've pre-computed from the square days. And let's plot the first 10 neurons from these square days. So here is a neuron that has a sort of spatial firing field along the border, the top border of the square environment, pretty consistently on all these square days. And here's another one that has this more loose field that's toward the top right corner of the environment. This one looks a little less stable, neuron 18. It has like kind of multiple fields. Maybe it's not the best placed cell. But you can see there is a variety of stability in where place fields appear across repetitions of the same environment with these long delays of 10 days. But nonetheless, I would like to start building a representation or a representational similarity matrix that we can use toward the ends that we discussed earlier, not get too lost in the weeds here. So the first thing I'd like to do is to build 3 different RSMs. The first one that we'll do is just going to take the average activity of the neurons on each day, like we already did, and then correlate the relative firing rates across all the days. So this is like if you had a 1 pixel rate map for every cell, right? And we're correlating those across all of our days of recording. So let's do it. First, not every neuron is going to be registered on every day. Sometimes neurons go silent, right? And we would just not see them in calcium imaging. So those neurons in these data will be NAND. So they'll be not a number on that day. So the way we can identify them is by identifying nants, right? So how I'd like to do this is iterate through every pair of days. So we have 31 days. So we could have these 2 nested for loops for day 1 out of 31 and then day 2 out of 31 to hit all of the pairs. And we'll grab the firing rates from these respective days from the mean firing rates that we calculated earlier. Okay? So a nice but not necessarily appropriate thing to do in this case could be if we just take all the firing rates, these average firing rates, and we add them together. If you add a nan with a actual number, a real number, it's going to be a nan. So we could add these two firing rates together and just say whatever's not a nan among these 2 days of recordings. Those will be the neurons that we've actually identified on the pair of days. So I call this reg mask because it's the registration that shows this neuron was recorded on both of these days. So now you have this mask to identify which neurons exist on both days. And I want to correlate the firing rates of all those neurons identified on both sessions. So we could use Pearson R from SciPy Stats. And we could say FR1 and just grab those cells that we registered on that day. FR2. Oh, live coding. This gives you 2 values in a tuple, and we just want to grab the first one, which is the Pearson correlation. Okay, I think this is right. No. Oh, there we go. Okay, it's done. That's quick. Yeah, so it just inverts. If you have a bunch of Booleans, it just like inverts it. Wow. We have 31 by 31 days looking at the similarity in the average firing rate of all pairs of registered neurons. So we see a lot of sort of dynamics. It looks a lot better on the computer, by the way, or the Crowdcast. But you see quite a lot of dynamic range. And it looks like maybe there is some structure. I promise there is some. You can see it here. But there is some dynamic sort of structure that maybe we could recover from this matrix or sort of investigate. For example, if you look especially on the Crowdcast, there's this sort of off-diagonal band. Can anyone guess what that would be driven by? There's this band of activity on day 11, and then it goes down the side here. Does anyone see that? You can't see it here very well, but it's right here. There's a band of higher correlations. What is that? So that's those are the days where the same shape has appeared again. So there's a higher correlation in the firing rates when the same shape is reappearing. Because remember how this experiment was designed. We have this sequence of geometries that repeats at a delay of 10 days. So maybe there's some meaningful structure in this RSM. But I think that we could get more specific and more detailed with how we're building this representation of CA1 activity. So we could use rate maps, right? We have them. So what you can imagine doing instead maybe is take cell by cell all the rate maps for these neurons, and whatever neuron we've identified on a pair of days, we could correlate that rate map from that same cell that we've tracked over time with itself. So you could correlate the rate maps cell by cell across all pairs of days. And we could ask, how similar are the rate maps of the same cells that we've tracked over time? When we change the geometry of the environment, if the brain's representation is very deformed, it's very different, then you'd expect low correlations in the rate maps, right? Because the spatial firing has dramatically shifted if the cells are remapping. Does that make sense? And we could take these correlations and fill out this giant matrix, and maybe it would have some more rich structure that we could investigate and compare to models. So let's do it. I did not task you with writing this function because there are some fail points, but you can look through this in more detail later. This is a function I've written that will build this rate map comparison matrix cell by cell for all the neurons that we've recorded. So I called it get cell RSM. So you can handle whether it's taking smoothed or unsmoothed rate maps. And what we'll do is we'll mask out the cells that were not registered, and we'll also mask out the areas of the environments that were unvisited on the pairs of days. So you can't really correlate the unvisited area with the visited area, right? And we'll iterate through the cells, cell by cell, and we'll iterate through the pairs of days. So comparing every cell's rate map to that same cell's rate map on all the pairs of days. So I just want to show you what this returns real quick. We say get cell RSM. We could say data animal. You want to get the rate maps, what do you put there? Remember? Maps. And determining whether it's smooth or unsmooth, that'll happen inside the function. But you could just throw this and it should work. Yeah. So there was probably all nan case in there somewhere, but it's iterating across all of the cells. There's 942 or something like that. I built it this way because we could do interesting things even with the RSMs of individual cells. Because you have a rate map for every cell. So you can build the representation cell by cell even if you wanted to, right? So I'll just show you what the shape of this is to give you that impression. So this is called cell-wise RSM now. We have 31 by 31 days and then 942 cells. So if you want to take the average of these similarities across all the cells, what you could do is average across this cell axis, right? But be careful because not all cells are registered on all days. So you need to take care of the nans. There's a function for that in NumPy, which is nan mean. So I'll take this cell-wise RSM, this representational similarity matrix based on the rate maps. And I'll say I want the mean to be across that last axis, axis 2. So I'll average across all cells. Okay, boom, done. Okay, let's see how it looks. Really rich structure. So the correlations become a lot more dynamic now when we're using the rate maps and the actual spatial firing of these neurons. So you get this strong off-diagonal band here at a delay of 10 days when the same environment is reappearing, right? So there is some level of stability with the same geometry being correlated in its representation with itself, right? You can also see a bit of a diagonal here, delay of 20 days. Are there any other sort of patterns people are picking out looking at this?
What about black lines, horizontal and vertical?
Yeah, so do you notice where the black lines are occurring?
Yeah, specific environments, I guess, not geometry.
Yeah. Yeah, so if I plotted this,
very different from the other ones.
yeah, there are probably some geometries that are very deformed in CA1. They cause very strong remapping. There are other geometries that are more similar to each other. So it seems like there's some reliable repeating structure in here. Okay, that's promising. And I think that suggests we could use the sort of rate map approach to build a representational similarity matrix that would allow us some model comparison. But I want to first show you one other way that we could do it that's very a la Gava. So we could instead, like, yeah. Yeah, so yeah, I'm super happy to answer that question. So the first one that we made, we were just taking the activity across a whole session, a whole recording, 40 minutes, and then just averaging the cell's activity. So averaging its firing. And so that's really, yeah, one cell every session, and we're just getting an average firing rate. The rate maps are the average firing spatially within the environment. So you have a 2D average firing rate on the space in the rate maps. So with the second approach that yielded this, we are building this representational similarity matrix, looking at our distances based on the rate maps in the spatial distribution of firing, instead of just the average firing across the whole space. So in CA1, it makes sense why this would produce so much structure. Okay, so let's imagine more similarly to what Giuseppe talked about earlier with the ePhys recordings, we could look at the activity among all pairs of neurons, maybe. And so we could take all of our cells from a given recording and then cross-correlate them with each other. So certain cells might have a very similar location or time at which they fire, certain pairs of cells, and then other pairs of cells would not be very related in their firing. So we could build this sort of coactivity matrix like this, where you have all of the cells being correlated with the activity of all other cells. And we could do this for different days and then match up all the cell pairs that we've registered across time. And we could take the upper or lower diagonal of this coactivity matrix and ask, how similar is the pattern of coactivity on all pairs of days? So if there's a strong change, there's a strong kind of change in the representation and the network activity across pairs of days, the coactivity among neurons would change dramatically. The relationship in firing among pairs of cells would change. So we could build the same matrix using this type of approach, because we have this sort of coactivity measure on every day. And we're able to track the same neurons. So we know if the same pairs exist on certain days. Okay. So let's do that. So let's take 2 example days. Here, day 14 and 24, which in Python is 13 and 23, which is a delay of 10 days, which means they're the same shape. So it's 2 kind of random presentations of the same shape that I've picked out of the data. So let's pick those days. And the way that you can correlate all cells with all other cells from a given day is we could say data animal, so that gives you the data for this animal. I can't remember if it's trace or traces. Let me cheat. Yeah, it's trace. Okay. And then we'll say target days. I'll pick the first target day. from that tuple. So this little line here is going to give you the correlation coefficients in a number of cells by number of cells matrix, this NP core coef function. Let's do the, I wish you could use Control Shift and Colab. We'll also build this coactivity representation for another day, or a second target day. And then down here, we'll take the triangle, or like the off-diagonal section of this coactivity matrix, and we'll correlate it on these 2 days. And I'll plot you the matrices. Oh, what did I do wrong? Oh, I'm probably missing something here.
Yeah, the Pearson control. Yes, we're missing a part.
Yeah, where did I miss it though? Is it in here somewhere? Oh, I think it's because target days. There we go. Okay, the issue was that I had some non-registered cells on one day that were registered on another, so I was trying to correlate NANs with not NANs. And it was like, what the heck are you doing? So, boom, you have this, well, you can't really see it super well there, but I promise if you look on the screen, you can see that there are certain pairs of neurons that you can kind of see them here, that have similar temporal patterns of activity. And there are many other neurons that have much lower correlations in their temporal pattern of activity, right? So these are the same geometries. We can correlate them and ask how similar is the pattern of coactivity. Here we get a Pearson correlation of 0.43. Pretty similar, the pattern of coactivity for a delay of 10 days. It's not bad. So let's try another day, a 14 and 25. So not the same shape, because it's no longer a delay of 10 days, which is the design of this experiment, right? So if you have a delay of 11 days, that's going to be a different shape. And you get a lower correlation for these two geometries at a delay of 11 days instead of 10. So we have 0.316. So maybe we could build out this sort of larger RSM comparing all the coactivity on all these different days with each other, and it could show some structure. I think there's some promise there. But I'm going to bin these really hard so that it just runs faster. These are some functions that I've written for simply comparing every day with every other day, and generalizing this coactivity comparison so that it works across all pairs of days. And you can read into that in detail if you'd like in the kernel. So, and you could also play with the bin size. So this is a temporal bin size. I promise you that the representational structure you'll recover is very related across different bin sizes. But I'm just going to do it at a very large temporal bin to save some time. In the meantime, let's get back to the presentation for a little. Okay, so while that's running in the background, the trace coactivity measure, I just want to briefly talk about how we could actually measure stuff explicitly that might be interesting off of this matrix and explore structure. So when you do this analysis across a bunch of mice, 6 mice, where we recorded for 31 days, you see this sort of rich structure and variable degrees across these animals. So you see in all of them this off-diagonal band with repetitions of the same geometry. You also see, as you move away from the diagonal axis, as you move further in time, the correlations tend to drop. This is something called representational drift, and we could go have a beer about that afterward. But you do see this sort of correlations drop over time. This is probably because the representation is changing with the animal's experience in the environment and just the evolution of time. But to kind of visualize the structure in the space, and here you can see these are actually the shapes of the environment on the axes of these matrices. But to visualize the structure, we could embed one of these matrices with multidimensional scaling or PCA or something like that and reduce the dimensionality. So let's do it. So here I've applied non-metric MDS, which you need to tweak the scikit-learn version, by the way. There's a bug. But I've applied non-metric MDS to embed one of these matrices in 2 dimensions, so you can see what the representation sort of looks like. And I've color-coded the day. So the darker blue is the first day of recording, and then the bright yellow is the last day of recording. And then I've drawn lines of the same between the same shape that's recorded at these delays. And so what you can see is there's this evolution of the representation over time. It's moving, it's drifting. But there's also an organization that's very consistent with how similar certain shapes are to others. So Giuseppe talked about earlier, maybe those dark bands are a really effed up version of the environment. It's very dissimilar from all the others. That would be this cross shape. And if you look in all these animals, this cross shape is often producing these strong dark bands. So it's a very deformed environment spatially in the brain. And there are other environments that are much more similar to each other. And in some sense, the similarities make sense. But we could also measure stuff directly off of these matrices by just sort of indexing out points of interest. For example, we could focus on one sequence of geometries. And let's say we want to compare every shape to one reference. So we compare every shape to the square as an undeformed sort of environment. We could look at how stable the representation is because we repeated the geometries at a delay of 10 days. So we could look in that off-diagonal section there. Or we could just look across all the repetitions regardless of the shape of the environment and see how the stability evolves with experience. So here's our framework. And when you look at the similarity to the square environment, you have the cross is a consistently deformed representation in CA1 spatially. And the donut shape and the rectangle are much more similar. And these orders of sort of deformity are preserved across repeated experience sequences 1, 2, and 3. If you look at the similarity of each shape to itself between the pairs of sequences, so 1 and 2, 2 and 3, you do find the stability increases in the later sequences. So as the animal becomes more and more familiar with all these variations in the shape of the environment, the stability of the representation improves, which is neat. And so if you measure this across that whole slice, you find this sort of continuous gradual increase in the stability at a delay of 10 days, which is cool. But are the effects the same if you don't use rate maps? Do you get the same measures and effects if we look at the trace coactivity? So I think the time is nigh to do that. So I've just iterated through all the pairs of days and did this coactivity-based analysis. And we get a relatively similar structure when we don't use rate maps at all. Rate maps have a strong spatial prior, right? Because you're assigning the activity to Euclidean space. But here we're just looking at the activity among pairs of neurons, however that's being generated by the animal's behavior. So we could try to ask this question by comparing sort of representations and the effects we get with this trace coactivity approach. When we do that across all the animals, we get a striking sort of semblance of some of the patterns we saw earlier. They don't seem quite as rich, and they seem to not have as big of a dynamic range in the correlations. And they're a little more variable across animals with how high the correlations tend to be. But nonetheless, there's a lot of structure that we could explore. Visually speaking, we still get certain shapes that are very deformed compared to a lot of others. They're very different. So the cross, again, is very distinct. And then certain shapes also seem more similar to each other, and we recover that drift, that temporal evolution effect still. So what about when we apply these targeted comparisons? Well, we get this rank order again. Certain shapes are more similar to a square reference than others. However, some of the ordering in the middle especially is a little different if you look at in detail at the differences. But overall, the extremes are relatively similar across the two approaches. We see this increase in stability again, which was surprising to us. And when we look across time, you see that experience in this environment that's constantly changing shape is improving the stability of the representation. So this is just summarizing those points. What I wanted to get to in just the last couple of minutes here, I know we're kind of risking going over time, is how we could use this approach to evaluate models, theories of hippocampal mapping. So going back to this sort of tic-tac-toe board, in like envisioning of the environment, instead of comparing the whole rate map of the environment or all the activity from the whole environment with every other entire environment, we could do something more detailed, a sort of finer grained set of comparisons. So we could break up the space into our tic-tac-toe board and instead compare the similarity of every partition within the space to every other partition, either on the same day or on different days. So we're comparing smaller subspaces in the environment. And the reason we might want to do this is because a lot of theories of hippocampus that we have at our disposal right now don't make a lot of claims about the effect of time and experience on mapping, but they make a lot of claims about geometry. So what we can do is do this sort of partition-wise comparison for every pair of days within every sequence. And each of these comparisons gives you 9 times 9 sets of comparisons for the representational similarity for every pair of days. So that's a lot of dynamics to compare against models, right? So every sequence now gives you all of this. And these lines here are just the parts of the environment that were not sampled on pairs of days. So for example, this corner is not sampled on the cross day, but it is here. But you can't compare those unsampled parts of the environment. So we'll just sort of block out those unsampled areas. Still gives us a lot of data to compare. We can build the same matrix with a model. So we could have a really naive model of hippocampal mapping and say place cells, all they do is they map allocentric space, sort of in cardinal compass coordinates, in a stable way over time. It's not changing. I don't know why you'd want to have a map like that. It's not very useful, but that could be one naive assumption. We could have another model that instead says the hippocampus cares a lot about geometry. And one way that it might care about geometry is it'll reset the firing location of a place cell after an animal contacts a boundary. So when it comes close to a wall and then it starts to run away from that wall, it'll reset the distance of the firing from that wall. So that's a boundary tethered model. The place cell dynamic is tethered to the boundaries. But there's other boundary-based models, like ones based on these funky cells called boundary vector cells. These are cells that fire at a certain distance from a boundary. They exist in the subiculum, for example, in hippocampus. They fire at a certain distance as an animal is running in a certain direction. We know they exist in the brain, and one claim could be that the place cells we see in hippocampus, they just take random weightings from boundary vector cells to encode where the animal is relative to boundaries. So those are three different models. What we can do is we could simply compare each of these theoretical matrices to the actual data and ask what model fits best. So when you do that with those three, you can't really see my noise ceiling here. You compare that to actual data, you get a rank order. The stability model does the worst in this experiment. So the hippocampus does, in fact, care about boundaries, the geometry of the environment. But of these geometric theories, the boundary vector cell model, the neurobiologically inspired model, actually does best. And it's starting to approach the noise ceiling, which is the theoretical maximum. That margin is the best that we could do given the noise in the data. So what I've tried to show today is that this type of approach, representational similarity analysis, you can leverage with neurophysiological data, like the data that Giuseppe presented earlier, or calcium imaging data, to actually look at representation in a really rich and informative way in these types of experiments where we can record over time and experience. And you can actually use these to compare against theoretical models with the RSA approach. And we can really benchmark our theoretical progress because there's more and more theories coming out all the time, but we need to actually test them against real data to know how good we're doing and to make improvements and move in the directions that Patrick was talking about earlier today. So with that, I'd like to thank the people that I worked with constantly on this project to collect these data. Alex Kyneth, who's starting her own lab at UIC in Chicago, Erica Gianferrano, who's a grad student at McGill now, and my supervisor, Mark. And I want to thank our funding sources, the Brandon Lab, and Unique and the main group for putting on an awesome educational session today. So, and I'd like to thank Giuseppe again for coming all the way from the UK. So, thank you.